In this slide, we're going to talk about what we call continuous conduction versus saltatory conduction. Saltatory conduction is when you have myelinated axons. The word saltatory actually means jumping or hopping. So the action potential actually can hop over the myelinated portions and jump from node to node all the way down the axon. So this neuron is myelinated. How do we know it's myelinated? These are the Schwann cells, and in between the Schwann cells are what we call nodes of Ranvier. This neuron is unmyelinated. Okay, so it doesn't have myelin, doesn't have Schwann cells. Notice the speed in which that action potential can travel. This situation, after 10 milliseconds, the action potential reached this far down the axon. Whereas if you compare it to this neuron, it's almost at the end of the axon. So what happens is the action potential, the sodium entering and the potassium exiting, only has to happen at the nodes of Ranvier. So it kind of jumps over large regions of the axon and gets down to the end much faster. So that's known as saltatory conduction using myelinated axons. And I'll remind you that the myelin is produced by oligodendrocytes in the CNS and Schwann cells in the PNS. And when we move on to the brain and spinal cord, we're going to learn that when you have a lot of unmyelinated neurons, we call that gray matter. When you have a lot of myelination, that's called white matter. Now, does it make myelinated neurons better than unmyelinated? No. It just depends on what that neuron is used for. All right. Sometimes the action potential has to be faster, but at other times it doesn't. So we don't need the myelination. Okay, next thing is we're going to kind of expand on this velocity, right? Velocities vary widely, all right? There isn't just one velocity. What determines velocity? There's basically the diameter of the axon. So some neurons have larger axons, and that allows them to transmit much faster. So that's one effect that changes the velocity, the diameter of the axon. The other one is myelination. We just saw that. The more myelination you can have, the faster the conduction speed. So what the myelin sheaths kind of act as insulation. Saltatory, remember, that's when the action potentials jump from node to node. And we call that saltatory conduction, which can increase the speed up to 30 times faster than if you did not have myelin. So we can classify neurons based on speed. We've got group A fibers are going to have large diameters and thick myelin. So they're going to be the fastest. All right. So they're going to be able to transmit that action potential very quickly. One example of neurons are somatic sensory and somatic motor fibers. So these are the fibers that can transmit, for example, pain from your skin to your brain really quickly. So we can perceive that very fast, but also we can control muscle very quickly. Group B fibers are kind of in the intermediate. They're not as large in diameter. They do have myelin, but they're found in more of the autonomic nervous system. And then we've got group C fibers. These are unmyelinated and small diameter. So those are going to be kind of the slower acting fibers. This shows us kind of white versus gray matter I mentioned previously. And we're going to look at, obviously, the spinal cord and the brain in upcoming chapters. But you'll notice that if we were to cut through a spinal cord, there's very distinct tissue differences. This out here is all the white matter. So that's going to be where we find a lot of myelinated neurons that kind of transmit up and down the spinal cord. Whereas this center area is gray matter. So it's unmyelinated neurons. The reverse is kind of true in the brain. The outer areas are unmyelinated, and then the myelinated or white matter is more deep in the brain. And these are actually examples of nuclei. Remember when I said nuclei are where you have kind of clusters and groups of cell bodies? And because cell bodies don't have myelin, those nuclei are gray matter. All right, so what I always find interesting is once you learn physiology, is there anything out there that disrupts it? And there's a number of things that disrupt action potentials. And one example are some of our local anesthetics. So we've probably heard of Novocaine and lidocaine. So Novocaine and lidocaine can be used to numb tissue, right? You might use it in dental procedures. They might do that if you're getting stitches. And how those local anesthesias work is they actually prevent the voltage-gated sodium channels from opening in your sensory neurons. So if you can't open voltage-gated sodium channels, you're not going to be able to transmit impulses to the brain. And those sensory neurons being pain neurons, right? You're not going to be able to transmit pain, and you won't be able to perceive when someone is stitching you up or pulling a tooth. So this is a great example of learning this complicated physiology and then applying it to a very clinically relevant thing such as these local anesthesias. There's also some things out in nature, some neurotoxins. So there's a lot of organisms that can be very nasty. All right, they can produce neurotoxins. So there's one called the poison dart frog. All right, this pretty little frog right here produces a toxin that actually blocks action potentials. Certain pufferfish and triggerfish produce neurotoxins that block action potentials. This guy's a scorpion, right? They produce actually a potassium channel blocker. Same thing, if you're mucking with sodium channels or mucking with potassium channels, you could interfere with how neurons are functioning. And then this is a snake, it's a black mamba snake, and they actually produce a calcium channel blocker. So a lot of our very toxic substances, their mechanism is based on blocking these ion channels that are required for neurons to function properly. Obviously, for me, I think the most interesting are the Novocaine and lidocaine, but there's a lot of other things out there that produce molecules that disrupt ion channels. All right, next thing is kind of a review. We've talked about a chemical synapse in the past. We called it the neuromuscular junction. This is just to remind you of what happens, and also to mention that it doesn't necessarily have to be a neuron to muscle junction. This could also be a neuron to neuron junction. So this is how neurons can communicate with other neurons. So again, this is reminding us about the importance of the voltage-gated calcium channels. The synaptic vesicles have to release neurotransmitter, and then the receptors here on this, what we would call a postsynaptic cell, or in this case they call it the postsynaptic neuron. These would be ligand-gated channels that open in response to the neurotransmitter, and then that might trigger an action potential or nerve impulse in this postsynaptic neuron. So that would be this neuron, the presynaptic neuron, stimulating this neuron, the postsynaptic neuron, to transmit an action potential. So this would be how neurons communicate with other neurons. Now it gets even more complicated because it's not a simple one neuron to one neuron situation. So here we have many presynaptic neurons influencing one postsynaptic neuron. And not all of these neurons are stimulatory. So we've got this idea of excitatory versus inhibitory. Excitatory is what we've basically been talking about, where a neuron could release a neurotransmitter. It causes depolarization, resulting in trigger zone, or threshold, and then causing an action potential down the trigger zone or in the axon. That would be excitatory. So you'll notice this neuron is excitatory, this neuron is excitatory, this neuron is excitatory. All right, so an excitatory postsynaptic potential is when you're trying to move that postsynaptic neuron closer to threshold. But what if you're inhibitory? We've got a lot of neurons that are inhibiting other neurons. So this neuron here, neuron 4 and neuron 2, are causing inhibitory postsynaptic potentials. That's where instead of depolarizing, they might be hyperpolarizing. Hyperpolarization will make it harder to get to threshold because you're taking that negative 70 and making it even lower. So how would you do that? Well, maybe you're bringing chloride in. Chloride. Chloride's negative. If you cause chloride to enter, that's going to bring that negative 70 down even more. Or you can move positive out. That would do it as well. So we want to make sure we understand the difference between excitatory versus inhibitory. Excitatory causes depolarization, which gets you closer to threshold. Hyperpolarization gets you farther from threshold. All right, so if we want to turn neurons off, we hyperpolarize them. Then there's this idea of a neuron has to take this all into consideration and then decide, am I producing an action potential or not? And that's called summation. And there's a couple types of summation. Spatial summation is where you might have two presynaptic neurons stimulating a postsynaptic neuron at different locations. Well, what could happen is they could kind of become additive. So you might get one of them stimulate and you just get this little depolarization. Then you get the other one stimulate and you get this little. But if they do it at the same time, right, if both of these stimulate simultaneously, you'll get threshold and cause an action potential. So if you've got different presynaptic neurons stimulating at different locations, that's called spatial summation. Temporal summation is if it's, one presynaptic just keeps firing, keeps stimulating over and over. So here it might stimulate once, then twice, then it starts stimulating very quickly, and that can add up to threshold. So you can reach threshold either by multiple neurons at different locations or a single neuron at a single location, but very quickly overstimulating, all right, to get to threshold. All right, so spatial versus temporal summation. All right, last video of this chapter is going to talk about neurotransmitters. So far, I think we've talked about acetylcholine, and that's about it. So we're going to talk about some other common neurotransmitters utilized in the nervous system.